Over the past decade, large language models have evolved from rule-based systems such as ELIZA and Sherdlu to the most advanced conversational models such as GPT-4 and Palm 2. Now there are many different applications for LLMs such as protein language models or Bloomberg GPT, combining AI and other fields. With technologies such as protein LLMs, some say that the future of LLMs lies in biology. While biology and AI used to be two very different disciplines, they are now being combined to find many protein structures that many human biologists could not. Welcome back to another episode of TechBites, in which we will discuss Alphafold and ESM2 protein LLMs from Google DeepMind and Meta. This is your host Sua, and let's get into it. Protein structures are made up of basic units that are easily computable. DNA is long chains of nucleotides that carry the genetic information of living organisms, which is used to synthesize proteins. The genetic information of every life form is saved in their DNAs using just 4 types of bases: adenine, cytosine, guanine, and thymine, ACGT for short. Having 4 bases means that the entire DNA structure can easily be represented in quaternary or binary, in which case computers can easily store and compute the sequences. Although DNA and protein are 2 different concepts, protein structures can be presented similarly as a 1D string of amino acids in a particular order. There are 20 different amino acids, and a protein structure ranges from tens to several thousand amino acids. With these 20 amino acids, the proteins fold in various ways to form 3-dimensional biological forms. There are expected to be around 80,000 to 400,000 proteins in the human body. Meanwhile, theoretically speaking, there can be around 10 to the power of 13,000 different proteins that could exist. This is an unimaginably large number and is even greater than the total number of atoms in the universe. Of course, not all of these combinations could result in biologically viable forms, but some could. The world's leading tech firms such as DeepMind of Alphabet and Meta are using AI technologies to find 3-dimensional folds of amino acid sequences. DeepMind has built Alphafold, a model that can correctly predict the protein's 3-dimensional shapes to within the width of about 1 atom. This outperforms all the other methods humans have ever made. Alphafold is one of the first to incorporate AI to unravel the secrets of protein folding, but it isn't built using large language models. Alphafold relies on multiple sequence alignment, MSA in short, which deduces the structure of a protein by comparing the protein sequence to evolutionarily similar ones. While Alphafold has significantly increased the accuracy of the prediction results, the process is still very computationally expensive and thus takes up much time. This is where Meta comes in. Meta has built ESM2 and ESMFold to predict the 3D protein structures. ESM2 is the large language model, and ESMFold is the associated structure prediction tool. ESM2 is as accurate as Alphafold at predicting the 3D structures of proteins, but it is up to 60 times faster. This is because ESM does not use MSA, and it is instead trained on millions of protein sequences provided by UniRef. Fed with an abundance of protein sequences, the ESM model was able to develop attention patterns. This is a highly used technique in natural language processing that is used to dynamically highlight relevant features of the input data and thus increase the accuracy. Unlike other large language models, the attention patterns in the protein language model represent the evolutionary interactions between amino acids and protein sequences. It also inputs only a single protein sequence and no structural information is needed. Here's a map of proteins provided by Meta called the ESM Metagenomic Atlas. It contains information on 1 million protein structures, from known structures such as the proteins in purple, or the lesser known structures such as the ones in yellow. Another advantage ESMFold has, as we can see from this protein map, is that it produces better predictions than Alphafold for orphan proteins that lack evolutionarily similar analogs. An exciting fact about models like ESM2 is that they can be reversed to produce novel protein sequences that do not exist in nature. Protein structures in nature comprise less than 0.1% of the theoretically possible combinations. Models such as protein MPNN and RF diffusion produce novel protein sequences that are not found in nature. RF diffusion is built using diffusion models, just like Midjourney and Stable Diffusion. They generate the possible 3D structure using a diffusion model, and then possible amino acid sequences are layered on top. Researchers are still developing better protein models, and the future of biological AI is promising. This episode discussed how AI models like LLMs and diffusion models are frequently used in different research and science areas. For more about how AI technologies are used in various fields of science, stay tuned on TechBites. This was your host Sua, and I'll see you in the next episode.